Welcome to the Molecular Devices Automated Electrophysiology webinar series. We're very pleased today to have two of our longtime customers present at this webinar. This is our 11th webinar in the series, and today we have our speakers are Dave Rock of Essen Instruments, or Essen BioScience, I'm sorry. He'll be talking about a sodium 1.8 assay, and then we have John Grace of Targacep talking about validation of the ionworks-videos with some nicotinic receptors. Dave, I'd like to turn it over to you, and we'll pause on your title slide, and then I'll go ahead and go through your bio quickly. So are you there, Dave?
Yep, I'm here.
Okay. I'll go ahead and hand you over the oops, sorry about that. I'll hand you the ball, and want to go ahead and briefly read your bio. So Dave has been at Essen BioScience for over 4 years, with his current position being Senior Project Leader of ion channel screening. His group is responsible for ion channel cell line assay development, as well as delivery of ion channel screening data, mainly with the ionworks Quattro and Barracuda platforms. Dave received his PhD from the University of Michigan Neuroscience Program, with thesis work on polyamine regulation of NMDA receptors using single-channel recording. Before coming to Essen, he was at Park Davis Pfizer for over 23 years in the CNS Discovery Department, working on ion channel and GPCR drug discovery projects. He was director in the CNS Biology Department. Dave, we're very pleased to have you here today, and I'll turn it over to you.
Thanks, Jim. I'm just going to jump right to the overview slide. So today, I'm going to talk about our NAV 1.8 assay on ionworks-Barracuda. And, before I walk through that, I'm going to talk a little bit about the corporate history around Essen and, our role that we've played in terms of bringing automated E-Phys platforms to the market. I'm going to talk about some of the challenges of NAV 1.8. Any of you that have worked on sodium channel assays, and I've been working on them for quite a while, NAV 1.8 has been quite a challenge to get a good, solid cell line that could be used for that automated platform. I'll probably only have, and I'm going to jump to that a little bit, only have a single slide to talk about how that was done, but it was actually a huge effort in our UK BioScience group. And talk a little bit about the challenges of doing sodium channel screening. It's really looking for state-dependent inhibitors and being able to do that in high throughput to drive projects. And that's where I come to the role of ionworks and the impact that it's made and how it's changed our ability to do drug discovery. Again, I already kind of mentioned it. We'll talk a little bit about cell line development. It's a single slide, but it actually encompasses a lot of work that was done. And then talk about some initial biophysical and pharmacology that we looked at on our NAV 1.8 currents on ionworks-Barracuda. Then move to the assay development piece, where I talk about a 2-pulse full inactivation protocol that we really use to try to see if the currents that we had with the NAV 1.8 cell line, we could get reproducible pharmacology and sensitivity with the use-dependent compounds. And then move on at the end to summarize some repeated gating pulse train protocol data that we've generated with NAV 1.8 to show you an example of the reproducibility within and between assays with the platform. I'll finish up with a summary and a few comments around future directions. Essen is a privately held company, Essen BioScience, a privately held company established in 1999 with a hybrid business model that involves the invention, manufacture, and distribution of instruments. I'll talk about that only. I have one quick point on the next slide about our InQSight live cell imaging platform, which is the main one that we market and sell. We also have a discovery services group that's focused on providing services on both our InQSight cell player assays as well as ion channel screening on the ionworks platform. And we're moving into an area where we're starting to consider doing collaborative drug discovery, and this is an area that, with the expertise of the senior leadership group, is something that's a natural for us in terms of moving toward. We're currently 75 FTEs, and discovery services is done on 2 sites, both in the U.S., in Ann Arbor, Michigan, as well as in Welland Garden City in the UK. Our team has extensive experience in both engineering and large pharma drug discovery that really is an interesting mix and a perfect fit for the kind of business that we're driving. Essen BioScience's roots is in instrumentation and drug discovery. Brad Nagel and Kurt Schroeder, the founders of Essen Instruments, founded a small company in Ann Arbor in the early 90s called Noveltech, which were the inventors of the flipper or fluorescence imaging plate reader that many of you use in your laboratories, and I know I did when I was at Park Davis. And it's a platform that's used for calcium imaging and doing some of the earlier optical assays for ion channel projects, and still a very useful tool for these projects. Back in the early, I mentioned that Essen BioScience was founded in 1999, and in 2001, Essen BioSciences invented the ionworks platform, what was called ionworks HT at that point, and licensed that instrument to molecular devices. This was the first planar high throughput voltage clamp electrophysiology platform on the market, and it's made a huge change in how we could prosecute these drug discovery targets. I mentioned that our instruments that we currently manufacture and are working on is the E-Phys platform, the long-term lifestyle imaging platform. And I won't talk about that a lot today, but there's a lot of information around the platform and its uses on our website if you're interested. In terms of background, sodium channels, voltage-gated sodium channels mediate regenerative action potentials that are really responsible for conducting information within cells through the activity of these ion channels. Electrical information is moved from one side of cells over very long distances in terms of neurons to others. There are multiple subtypes of sodium channels that have differential expression found in the nervous system, heart, and muscle cells, and have different biophysical properties and sensitivity to a number of pharmacologic agents, and specifically tetrodotoxin has been used to differentiate between 2 broad classes of sodium channels. There's the large alpha subunit, that's the pore-containing subunit, that's in most cases over 2,000 amino acids. And many of the compounds that we talk about that are of interest as sodium channel blockers interact with this alpha subunit. But in addition, there's an auxiliary beta subunit that really affects the translocation and stabilization of the alpha subunit in the membrane and affects the biophysical properties when co-expressed with the alpha subunit. In order to get sodium channels that mimic the neuronal and cardiac channels, alpha subunits must be present. Beta subunits must be present. NAV1A, which is also known as PN3 a long time ago, or currently SEN10A, is a subtype of TTX-resistant channels and is located primarily in pain-sensitive DRG neurons. Antisense and knockout experiments that reduce the levels of NAV1A in preclinical animal models support NAV1A as a target for novel analgesics. But one of the challenges of dealing with NAV1A is that robust, stable expression of these channels in the mammalian expression system has been a challenge. And it's been a challenge to get the expression to the level and low cell-to-cell variability such that they can work in some of these higher throughput automated platforms. it often requires overexpression of the auxiliary beta subunits. Unlike some of the other channels, like NAV1.5 and 1.7, you don't have to overexpress auxiliary subunits in, for example, HEK293 cells in order to get large sodium channel currents. This isn't really the case with NAV1.8. So it's a challenge in terms of working with this target. Now, sodium channel blockers actually have a very unique profile. They interact with a certain state of the channel, and this state is an inactivated state. And let me back up a little bit. When the membrane potential is at very negative potential, these channels are in a closed state. And upon depolarization, wrong direction. And upon depolarization, channels move to the open state where they conduct current. You can see this in one of the traces here. It's just an example trace below where it's not a very high resolution. I'll show some higher resolution in some of the slides coming up, where this large inward deflection is due to the opening of sodium channels. That opening doesn't occur for very long. In fact, in terms of the average open time for a sodium channel, it's in the 1 to 3 millisecond range. And upon continued depolarization, the channels then rapidly move to the inactivated, the wrong direction again, the inactivated state. Use-dependent or state-dependent inhibitors, which all of the listed compounds or the approved compounds that have a sodium channel mechanism have this type of activity, where they bind with higher affinity to this inactivated state of the sodium channel. These include anticonvulsants, analgesics, and local anesthetics, as well as class 1 antiarrhythmics. These compounds show this state or use-dependent profile, which can be demonstrated in voltage clamp experiments. These traces are actually from one of the first papers with the ionworks platform, and I believe it's a NAV 1.3 cell line that was used in these experiments, where if you do a repeated gating protocol where you activate the sodium channel here 5, 6, 7 times, you get currents that have a certain amplitude. The amplitude of this current is proportional to the number of channels activated, and before drug is added in this control well, we get a fairly stable amplitude of current. In the presence of amitriptyline, which is a tricyclic antidepressant, which actually shows this use or state-dependent profile of sodium channel block, you see that the first pulse in this train is blocked about 50%, but as we go down, and as we repeatedly open and close the sodium channel and move it into this inactivated state, the amitriptyline binds to those sodium channels and blocks almost all of the current, or a high amount, 80 to 90% block by the end of this short pulse train. Other compounds, and I mentioned tetrodotoxins, are state-dependent inhibitors, and you can see the difference here in this profile, where the control well looks very similar to the well with the amitriptyline treatment, where you get a stable current over multiple pulses, and when 1.2 nanomolar of TTX is added, it reduces the current about 50%, and the repeated gating causes no increase. Pathological states that include chronic pain and epilepsy, as an example, lead to long-term depolarization and increased activity of sodium channels. So these use or state-dependent compounds selectively target cells that are showing this pathological activity and don't cause huge effects on the normal physiologic activity. It's a challenge to screen in this way. Optical assays, the control of the different states of the channels are variable. It's hard to know whether the channels are in closed, open, or inactivated states. The way to do this is with electrophysiology. And while manual electrophysiology, you can do these kind of experiments one at a time, the biggest difference that happened with the introduction of ionworks platform was that you were truly able to do those kind of studies in a way where you could support SAR for a project. It's true plate-based voltage clamp pharmacology. And I'll be showing you examples of this with voltage-gated targets. I have a little bit of data from our Quattro system, as well as the ionworks Barracuda. John Briggs is going to be talking in a few minutes, and he will talk about the same kind of plate-based pharmacology for ligand-gated ion channel, the nicotinic channels. The one thing I'd point out is that the instrument is valuable for reagent or cell line generation. I'll talk about a dilutional subcloning experiment that was done, and we chose a subclone of our initial pool that had higher level of expression. We did that using the ionworks platform and doing assay development using pharmacologic tools of interest. It's developing not just an assay based on the biophysics of the channel that you're dealing with, but one based on the pharmacology. I'm going to show you examples of some pharmacology, but as customers come to us with a unique problem with a compound that has certain kinetics, we can change the protocol to tune the assay to be sensitive to that novel class of compounds. The throughput allows screening, which I would call a targeted HTS, not the million compound screen, but focused library screens. It also can provide hit to lead, as I've kind of mentioned and tried to talk about, as well as supporting SAR for medicinal chemistry. You can do subtype selectivity, off-target pharmacology, and with the advent of the Barracuda, and I'm not going to talk much about it today, you can do experiments that are very similar to the ones that you do on manual patch clamp, on the manual patch clamp platform, but do them in very high throughput in a way that you can drive pharmacology with protocols there. And I'll show one quick example of where we could do multiple holding potentials and do pulse strains from those multiple potentials in some of the subsequent slides. Now, you can cut throughput in a variety of ways, but what I like to be able to say is based on the kind of 384-well format of the Quattro and Barracuda platforms, that we can test hundreds of IC50s per instrument in a single day, and thousands of single point determinations per instrument per day. It allows ionworks to be able to support project progress from hit to lead identification to efficacy IND type studies. So this slide just kind of summarizes our basic solutions and metrics that we use for the ionworks NAV 1.8 assay. And the cell line that we used was licensed in from the University of Michigan, and the background line is HEK293, stably co-expressing human SCN 10A NAV 1.8 and a rat beta 1 auxiliary subunit. We use cell suspensions in our ionworks assays that are a bit higher than what some groups use, 3 to 4 million cells per ml. We think this is critical in terms of being able to have stable, high-resistance fields, particularly in the population patch PPC mode. The external solution used in the experiments is a HEPI's buffered sodium chloride-based solution with full-strength sodium, 137 millimolar of sodium chloride. The internal solution is a mix of potassium gluconate and cesium chloride in a HEPI's buffered. The cesium chloride is added in this assay to block potassium channels, endogenous potassium channels that are present on HEK293 cells. First rating agent is amphotericin, 120 micrograms per ml. Fluid additions are done from the high position in Barracuda with a mix. We did this initially to mimic the way that compounds are added in ionworks Quattro with 3 to 5 minute incubations for the studies that I'll talk about today. I'll make a point to come back to this later. This is something that we think we can change the way that we add compounds to help with some hard to deal with compounds as well as some of the assay stability, enhanced assay stability. Data analysis is fairly simple. We measure the peak inward current, and here on the right-hand side of the slide, I have a raw data trace from a single recording in a well of ionworks Barracuda. The sodium channel current is an inward current, a downward deflection here in blue. We measure the peak value for the inward current within a window of probably around 10 milliseconds between the beginning and the end of the voltage clamp step. Baseline is either determined before or after current has been inactivated during the test pulse. So I mentioned that it's important to get robust expression in order to get good solid ionworks assays. And so our UK group did a lot of work in terms of subcloning to maximize that expression in our NAV 1.8 line. All this work was supported with the ionworks Quattro in terms of analyzing these potential subclones. The initial clone that we obtained from the University of Michigan, we look at a couple of metrics to try to determine, and these are all done from single hole recordings. So each circle here in red for the initial clonal pool represents the amplitude of the inward current in a single cell. And in terms of the average amplitude of currents in successful recordings that cells that had greater than 0.1 nanoamp of current in the initial pool was 27%, and the average maximal current when we put together the average of all cells expressing was only 183 picoamps. Probably not good enough to do the kind of experiments that we would want to do on our ionworks Barracuda platform or Quattro platform. There was an extensive subcloning effort, a dilutional subclone where the group in the UK analyzed the data from 200 subclones that they evaluated on ionworks Quattro. From those subclones, they identified clone 189 that had a higher percentage of cells expressing, 55%, and had a higher average current of about getting close to 300 picoamps. But even that level wasn't really adequate for what we wanted to do. One of the things that's kind of interesting in clone 189, I know we looked at it here in the U.S., is that there were some outliers that kind of point to this one cell that was out at 1.5 nanoamp current. It suggested to us that, there maybe was something special we could do to increase that expression. And so we worked at a variety of different optimization protocols and found that a combination of some cell handling as well as, very importantly, holding at negative potentials for long periods of time really enhanced the amplitudes of the currents to the point where 75% of cells expressed greater than 0.1 nanoamp, and the average current in those cells expressing was getting close to around 600 picoamps, which really is in the range for a potential assay in ionworks. One of the problems that we had with trying to do these kind of experiments on ionworks Quattro is that 5 minutes, because of the unclamping that occurs between evaluating different quadrants in the patch plate as well as doing a fluidic addition, it made that 5 minutes holding at -90 made it not very amenable for doing an assay on the Quattro platform. So that was probably toward the end of last year when we decided to move this assay to the Barracuda. So what I'm showing in this slide is a biophysical characterization of NAV118 currents on Ionworks Barracuda, and all these recordings that I'm talking about now are done in the PPC mode, where each well is currents from each well are the average of up to 64 different individual cells. So the current amplitudes here in an IV plot, when we look at the maximal current in this one well, was around 450 nanoamps, picoamps. And so when we constructed an IV curve using the whole plate, data from the whole plate, all 384 wells, we got an average current of around 400 picoamps with a fairly tight distribution. The threshold for activation of the current was around -40, peaking to between 0 and +10 millivolts, which is fairly in alignment with the literature from manual recordings of NAV 1.8. One thing that noted is that the activation and inactivation kinetics were slower than NAV 1.7 or some of the other TTX-sensitive currents, and this is consistent with the literature as well. The traces here are an overlay of a single well recording from NAV 1.7 in red, which you can see at faster activation, faster inactivation than NAV 1.8 in blue. Show slower to activate and slower to inactivate. Again, consistent with the literature. Inactivation measured on Ionworks Barracuda and PPC recording showed similar data, whether we use the 1 second or 5-second pre-pulse at different holding levels to inactivate the channels with a voltage of around -50 millivolts for half inactivation of the current. Again, consistent with literature. The hallmark of a NAV 1.8 current as compared to the TTX-sensitive current is that it shows a relative resistance to blocks with TTX, but it's still sensitive to the local anesthetic compound, and here the prototypical compound is Tetracaine. On the left-hand side, we have data from a single well where in control we have about an 800 picoamp sodium channel, NAV1.8 sodium channel current. This blocked completely, shown in red, with the addition of 300 micromolar Tetracaine. 3 micromolar TTX in another well on the same plate showed almost no block of the current. This level of TTX would block the, for example, NAV 1.7 completely. Vehicle controls done on the same plate show very little block. So the blocks that we see with Tetracaine aren't due to vehicle effects, as the recordings were very stable. The next thing that we did, and this was actually pretty early on, is that it was important to evaluate the stability of these currents over time on the platform. In order to do this, we used a protocol that we wouldn't use necessarily to do pharmacology. We held cells at -90 and then did 20 millisecond steps to +20 millivolts at 0.5 hertz for a 30-minute time period. And what we did this to evaluate this period here was right after the addition of amphotericin. So we're monitoring access here, where we get increased current size over time, and then a stable period that lasted about 10 to 15 minutes. When we look at longer time periods, we start to see fall off of the current, and this is the average current of all 384 wells of the plate. Field resistances increased slightly in that same time period where we were gaining access, and were stable out to probably around 20 minutes where they started to fall off. It's really important to know for when you're going to do your pharmacology, where is the optimal assay window to do your pre-compound evaluation, and then how much longer will the recordings hold on in order to get a good solid measure of pharmacology. And in this assay, we felt very comfortable going out to about 15 minutes. So that was the data from, an average data from a whole plate, but it's also important that there's not a lot of well-to-well variability. So this is a current amplitude distribution of recording of NAV 1.8 at the time at which we would do a pre-compound measurement. The distribution is fairly tight, with around a 12 to 13% variability. This is the mean plus or minus standard deviation of 382 wells. It's also important that the currents be stable following compound addition, and 0.3% DMSO vehicle. We saw a slight run-up, and we see this in our sodium channel assays, here in this case, about 10% run-up between control reading before adding drug and then a 5-minute incubation of vehicle. So stable amplitudes. The other thing that's very important in terms of evaluating your cell line as well as performance of the machine is that this profile that we show when you're looking at field resistance, and these are average field resistances for the plate over time, is that from the beginning when cells are first added in this experiment, we saw a field resistance of about 72 megohms, an average field resistance of 72 megohms, and over the course of when you get access as well as just in time, we saw an increase in field resistance to a level of around 89 when we did our first pre-compound evaluation of currents, 89 megohms, and there was a minimal decrease in field resistance following a fluidics addition that occurred at the 10-minute time point in this assay. All important things to evaluate in order to make sure that you're getting good, stable recordings of NAV 1.8 currents. The next protocol that we decided to move to is what I refer to as a 2-pulse full inactivation protocol. And it was designed to be able to evaluate resting channel block as well as an activated state block within the same protocol. To do some of the initial experiments, we used a multi-protocol mode in ionworks where we look at different scans with what I'll refer to as different delays between the test pulses. And I'll walk through this and explain what I'm talking about. In this protocol, we hold at minus 100 millivolts, and then what I refer to as test pulse 1, we step for 30 milliseconds to plus 10 millivolts to evaluate the amplitude of resting channel, full activation resting and resting block after we add compounds. We use a 10-second depolarization to 0 millivolts in order to inactivate the channels and allow, when drug is present, for the binding of compounds to the inactivated state of the channel. In order to make a measurement, we have to drop back down to minus 100 millivolts for in this case, varied amount of time in order to get a current to measure. In control, that represents the number of channels that have recovered from inactivated states. We use a delay of 200 to 500 milliseconds, and I'm kind of jumping ahead, to get current amplitudes in control that allowed us to do pharmacology, but we wanted to make sure that we didn't have time periods too long to allow compounds to unbind from the inactivated state. This protocol is really tuned for some of the higher affinity compounds that groups are working on now as sodium channel blockers. The local anesthetics, while tetracaine works in this protocol, and you'll see data from that, it's probably not the best protocol to evaluate some of the lower affinity state-dependent inhibitors. One of the things that we were very interested in, though, was the stability of these recordings from run to run and within plate. You've seen examples of what the variabilities look like within a plate, but this plot that I have over here on the left-hand side of the slide represents field resistance and current amplitudes for 6 different runs done on the same day with different recovery periods in terms of this delay, and we find very consistent data. And when we averaged over 19 runs, the average current amplitude of test pulse 1 was 0.53, 530 picoamps, and with a fairly tight range. And the fractional recovery of channels with a 200 millisecond delay pulse here was around 0.44, and 500 millisecond fractional recovery gave us about 6.62 fractional recovery in 12 different experiments. Fairly consistent data. We then wanted to look at tetracaine block of these currents. And so the tetracaine block, and this is data from a single well, a single group of wells, 10-point concentration response curve for tetracaine. And what we see is that tetracaine under rest, and test point 1 represents resting conditions. Tetracaine had an IC50 of around 20 micromolar, 25 micromolar, independent of the delay, because that's not really part of test pulse 1. But in test pulse 2, it showed a more potent block, a block of the inactivated state of the channel with a 400 nanomolar IC50 with a 50 millisecond delay. And as we increased that from 50 to 500 milliseconds, it went out to 1.2 micromolar, but fairly consistent block. As expected, as that delay period got longer for the compounds like tetracaine, there is a much more rapid recovery from inactivation. The other thing we wanted to see is what the Z prime or screening window would look like in this kind of assay. And this is a pretty complicated slide. I'll walk through this quickly. There are 4 conditions that we tested, and we tested each of these conditions in a column where we looked at high concentration tetracaine block, 300 micromolar, a mid concentration of tetracaine for test pulse 1, which is around 30 micromolar, a mid concentration of test pulse 2, which is around 1 micromolar, and the low DMSO is a DMSO vehicle control. To evaluate screening window, you look at the difference between the average block of 300 micromolar tetracaine along with its standard deviation and the vehicle control. And when we do those calculations in this plate, it's around a Z prime of around 0.84 for test pulse 1. And you can see that the way the plate was set up, they were done in sequence where column 1 had high, column 2 mid, test point 1, mid concentration, test point 2, and column 3 low in column 4, and then repeated that throughout the plate. One of the things that I look for in an assay is how tight the IC50-like block is. And so for test pulse 1, we used a 30 micromolar concentration and got about a 60% inhibition, or around 40% of control, and a fairly tight distribution for test pulse 1. The same kind of data, we saw the same kind of effect in the other, at the other test pulse, which was done at a 500 millisecond delay. And again, very tight distributions and tight IC50-like block. When we calculated Z prime for that for test pulse 2 is around 0.8. We replicated this plate 3 times on a day and saw these same kind of Z prime values in the same range. Low variability around midpoint for that mid concentration, that IC50-like concentration, a very tight assay. We had similar data that I don't show here for the 200 millisecond delay as well. Being conscious of the time and trying to move along a little bit here, we have a little bit more data to talk about. The other thing we did using this protocol then is to look at replicates of Tetracaine block within a single plate to know what our intraplate variability really looks like. And so this is an overlay up here in the left-hand corner of a Tetracaine concentration response curve, 8 of them overlaid from individual wells. Each point represents data from an individual well for test pulse 1 and test pulse 2. You can see that the data are very consistent with Tetracaine between runs, where the geometric mean of these 4 or 8 IC50s is in the 20 micromolar range, with one of the runs being around 15. And then when we did 8 within a given run, you can see that the range of IC50s goes from 17 to the low 20s, 23, 20, 24 micromolar. Very tight data. The data with test pulse 2 was more complex. Most of the time we use a single site fit, and this is a fit of the resting pulse test pulse 1 block, which fits the data very well. But I know when I showed these data to my colleague in the UK, Tim Dale, he looked at it and he said, you know, this looks like a classic 2-site fit, not 1-site. Although it's kind of the 1-site fit does a okay job describing data. When we fit the same data with a 2-site fit, we saw 2 different components. One of them that had an affinity that was much like resting block, and that was around 33% of the fit, and then a high affinity, which we would have expected Tetracaine to be higher, and you'll see Tetracaine data from pulse train, which is more in this range of around 100 to 200 nanomolar IC50, and that was the majority of the fit. So a fairly complex pharmacology using this test pulse 2 with these lower affinity compounds that have that probably do reverse off in the channel during the delay period. The next couple of slides, I'm going to just talk about some of the data from our pulse train protocol. And the first experiment that we did here, I kind of labeled this as extending what's done in ionworks Quattro. In these kind of protocols, we look at repeated gating of the sodium channel, like I mentioned early on in the presentation, where we activate the channel at a 10 hertz frequency with 25 pulses, each of them 30 millisecond steps to 10 millivolts. And what we evaluate for the most part in these kind of experiments is the IC50 for the first pulse, which represents kind of the resting state block, and then the last pulse, the 25th pulse, which represents the use-dependent or state-dependent inhibition with the compound. And you can see that in this plot, and this is again data with Tetracaine, each symbol represents an N of 4, but you can see that when we hold at minus 100 millivolts, which were very few, if any, of the channels are in the activated state at that point, we get this 20 micromolar, well in this case it was 8 micromolar IC50 with Tetracaine, and then the use-dependent effect at the 25th pulse, way down in here in the squares, the IC50 in this case is 260 nanomolar. So a very profound use-dependent block. This would represent one experiment in Quattro. In this experiment, we repeated the pulse train from these 5 different holding potentials: -100, -90, -80, -70, and -60, and could show this state-dependent block in all cases, and show that as we change the holding potential and started to inactivate channels from -100 to -60, which should represent about 30 or 40% of the channels in the inactivated state, we see a much more potent block of even the first pulse in this experiment. These kind of experiments are possible, but only useful when you have lower affinity compounds, because we did do some experiments with higher affinity blockers, and once you achieve the block at the -100 potential, the subsequent blocks at -90, -80, and -70 were greatly effective. So it's probably a protocol that could be used to evaluate a large number of compounds, but only those with lower affinity. In addition to that state dependent, we then looked at a number of local anesthetics as well as amitriptyline in terms of our validation. We wanted to know how, with this pulse training protocol, what was the variability like within runs. You can see the same kind of state-dependent block in here. I'll just go over this example of tetracaine with a more potent block of the 25th pulse. I'm afraid these are all inverted. I got my color scheme wrong in terms of the legend. The 25th pulse block is more potent compared to the first pulse block. IC50 for the first pulse is around 12, and 300 nanomolar for the 25th pulse. Each point on this curve represents a mean plus or minus standard deviation of an N of 4. And so the data within the plate runs are very tight. Bupivacaine, mexiletine, amitriptyline, and lidocaine showed a very consistent state-dependent, use-dependent block, consistent with the literature, low standard deviation in the replicates within a plate. And the throughput in this assay would be around 30 compounds, 10-point concentration response curves with reference compounds, full blocking, and vehicle controls all done in a single run. It's also important, so these are consistent within runs. It's also important to have low run-to-run variability. And I'm not going to try to walk through all the data in this slide, but these are the IC50s for those same 5 compounds in the first and 25th pulse done in 4 individual runs. The data you've seen on the previous slide was from run 4. And when we look at the average IC50 values, the values from each individual run are within 2-fold of that average. So it's a very tight, low variability between runs. In terms of summary, I think I've showed you data that we have a robust, reproducible assay, NAV1.8 assay on Ionworks Barracuda, and we can use multiple protocols to evaluate drugs. There's an excellent screening window for single-well, single-concentration screening. This could be done with either the 2-pulse full inactivation protocol or the pulse train protocol. And it's a good example of plate-based voltage clamp pharmacology with a throughput that allows you to evaluate state or use-dependent inhibitors of NAV1.8. In terms of future directions, there are some issues with how compounds are added, and we all know that there's been a nice demonstration of how certain lipophilic sticky compounds you can get potency shifts if you do just single additions. So we've been working more recently with some multiple addition protocols from different heights and doing it in different ways that we think will maximize the exposure of these compounds that are hard to deliver, as well as potentially enhance assay stability. Another direction that we're going to be going with sodium channel assays on Barracuda is to expand our offerings out to a full panel of sodium channel subtypes. Currently offer NAV 1.5, 1.6, 1.7, and 1.8 in pulse strain or full inactivation protocols. And we will be validating in the next coming months NAV 1.1, 1.2, 1.3, and 1.4. In terms of acknowledgement, I talked a lot about our UK group. I'll just first mention that Vince Brophy was the one that licensed this cell line in from the University of Michigan. Our UK group, which includes Srini Kanamuli, Sandra Alpino, and Susanna Alcantara, were really involved, along with Tim Dale and Del Trezise, in terms of doing the subcloning and improving the cell line to make it one that can be used on the ionworks platform. Kevin Allard, Livia Pekka, and John Rauch were responsible for doing all the cell culture and recordings that you've seen on the ionworks platform. Laszlo and Kirk are supportive, as always, of these kind of efforts. So with that, I'll entertain questions.
Actually, Dave, we're going to hold the questions till the end of both speakers. So thank you.
Okay.
Very nice talk. You're really getting nice results with your sodium channels. John, I'm just unmuting you here, and I'll go ahead and I'll hand this over to you, and then I'll go ahead and go through your bio. As I mentioned, we are taking questions. Please use the Q&A tab on the WebEx. I'm just handing it over to you, John.
All right, can you hear me, Jim?
Yeah, I can hear you. And go ahead and get your slides up, and then I'll go ahead and read your bio while you're doing that. So John received his PhD in neuroscience from Wake Forest University, where he focused on acquired changes in ion channel expression and function that can result in hyperexcitable states underlying pathological conditions such as epilepsy and alcohol withdrawal syndrome. Currently, he holds the position of staff scientist in the Electrophysiology group at Targacept. It's a clinical-based biopharmaceutical company in Winston-Salem, North Carolina, that develops small molecules targeting neuronal nicotinic receptors for difficult-to-treat diseases and disorders of the nervous system. He's also the project manager for Targacept's Parkinson's disease program, where he helps coordinate the preclinical processes of drug discovery, compound characterization in vitro and in vivo efficacy, and compound development for potential Parkinson's disease therapeutics. In addition, Dr. Grace serves as adjunct assistant professor in the Department of Neurobiology and Anatomy at Wake Forest University. And John, I can see your slides, so you're up.
All right. Well, thanks, Jim, and thank you for the invitation to participate in today's webinar. So as Jim mentioned, I'm going to talk today about some validation data that we've gotten from the Barracuda system using nicotinic receptors. And I'll just sort of jump into the outline. I'm going to give a little background information on Targacept. Also, I'll go over the introduction and sort of the goal of the study that I'm going to be showing the data from today. I'll also talk a little bit about the Barracuda methods we used and focus a lot on the data analysis that we performed. I'll then show the validation data at the end and kind of give a brief summary at the end of the slide presentation. So as Jim said, I want to acknowledge this work was done while I was a member of the Electrophysiology group at Targacept, and as he mentioned, Targacept is a clinical stage biopharmaceutical company in North Carolina with a platform to develop molecules that target nicotinic receptors. So nicotinic receptors are located throughout the brain and the body, but in the brain they're primarily composed of alpha and beta subunits. They are pentameric ligand-gated ion channels with either a homomeric subtype such as the alpha-7 receptor or heteromeric subtypes such as the alpha-3-beta-4 and the alpha-4-beta-2 subtypes, of which I'll show the data today on. They're located primarily presynaptically, controlling the release of neurotransmitters from the synaptic terminals. There is also some evidence of them being postsynaptic, involved in volume transmission and fast synaptic responses. So just to sort of introduce this study, as we all know, with the recent development of these high-throughput automated electrophysiology-based technology coming out, that's able to do this high-throughput screening, we need to validate these tools and make sure that they're producing data that's similar to sort of the gold standard manual patch clamp electrophysiology methods that we all know. And one of obviously the most recent introduction is the Barracuda. And so the goal of this study, we wanted to compare the current response properties of the alpha-3 beta-4, human alpha-3 beta-4 nicotinic receptor and human alpha-4 beta-2 nicotinic receptor to their endogenous ligand acetylcholine using three separate platforms. First, we want to look at manual electrophysiology using our Dynaflow system, which is a product of a company called Tetracaine, a Patch Express automated electrophysiology platform, which is a higher throughput. 16 simultaneous recordings in gigaohm seals can be achieved on that platform. Then, of course, the ionworks Barracuda, which is a truly high throughput automated E-Phys platform where we can record from up to 384 wells simultaneously. And I do want to mention that all the data that I'll be showing today has been published. Here's the reference for that publication that I've given at the bottom there. So just a brief introduction on the Barracuda. I know most of everyone listening is familiar with this, and Dr. Rock gave a nice introduction as well on the Barracuda system. But just to reiterate a few points, again, it's a 384 well assay plate that has what I think is a nice aspect of this is 384 discrete amplifiers, allowing for recording of ion occurrence from all 384 wells simultaneously. And again, using this population patch clamp technology under perforated patch conditions allows for higher quality data and allows for up to 64 cells per well in each of the 384 wells. So I'll just sort of jump into what methods that we used for our Barracuda experiment. So we used two separate cell lines. The first being the human alpha-4 beta-2 receptors, which were stably expressed, and a Schett cell line. Also the human alpha-3 beta-4 receptor, nicotinic receptor, which was stably expressed in CHO cells. We used trypsin to harvest these cells, and we did resuspend them in the external solution before placing them into the ionworks Barracuda. And there's our recipes, show our recipes for both the external solution, sodium chloride-based external solution, and an internal solution, potassium chloride-based internal solution. And I did want to point out that we were primarily using Essen as our perforating agent, 1.3 milligrams per 100 milliliter internal solution. This was added for a 10-minute period to gain access to the cells, and just to spend a few minutes on why we went this route. So beta-Essen has been shown to be an effective agent for perforated patch experiments. I think back in 2004 was the first publication showing that beta-Essen can have higher success rates with less rundown as compared to amphotericin. And a more recent publication showed that using the ionworks Quattro automated platform, there really was no difference between beta-Essen and amphotericin. Some advantages to this perforating agent, the beta-Essen perforating agent, is that it can allow larger molecules such as ATP through the membrane to help prevent rundown. It's also water soluble and is cheaper than amphotericin, so that's always a plus. It is light sensitive, so there are precautions that need to be taken into consideration as well. But this perforating agent worked well for us. So this is an example screenshot from one of our runs with the alpha-4 beta-2 nicotinic receptor on the ionworks Barracuda. And just to orient you on our setup was we had a no compound control in the first column here. Increasing concentrations of acetylcholine was added from columns 2 through 12, and then this same similar setup was repeated on the right-hand side of the plate. And so just showing you some example raw traces, a lower concentration here. From the blue box, we have a little bit of current from this lower concentration of ACH, and at the highest concentration of acetylcholine that we use, we can get up to 1 nanoamp current with the alpha-4 beta-2 receptor cell line. This is an example run for our alpha-3 beta-4 cell line. Again, similar plate setup with the no compound control and increasing concentrations of columns 2 through 12, and similar repeat on the right-hand side of the plate. And as you can see, again, smaller responses from lower doses of ACH, and with that maximum acetylcholine concentration we used, upwards of between 1 and 2 nanoamp current. So we were getting nice current responses and nice dose responses for both the alpha-4 beta-2 and the alpha-3 beta-4 nicotinic receptors. And so I'm going to spend a few slides, a few minutes on going through our analysis methods that we used. And we did not use the software, the analysis software that came with the Barracuda system. We actually exported all of the raw traces and the raw seal tests as CSV files, and then put them through our custom algorithm that we wrote in MATLAB to basically line up the compounds and the concentrations that were used, pull out any types of measures or current metrics that we thought were valuable. Not only were we looking at amplitude, but such things as charge, time to peak, the decay of the response as well, and organize all those responses and print out some dose response graphs, such as shown here on this example run where we were doing a validation run with acetylcholine on the human alpha-3 beta-4 receptor, and just putting out all the relevant EC50, EMA-XL coefficient information from these dose response curves. So I want to walk through how exactly we went through analyzing this data and what we did to come to those dose response curves. So here, again, here's a screenshot of an example run on the ionworks Barracuda. The first thing we did is we would highlight all the wells, right-click, and then export the well trace data. And we would do this for both seal test 3 and for the compound trigger, the actual current responses as well. So we would export the well trace data for both the seal test and the compound trigger, which would give us 2 CSV files, data files that we would then put into this MATLAB algorithm. And we also did similar sort of filtering exercises to try to get rid of wells that poor quality wells, wells that didn't have where no seal was present. These were wells that were typically less than 20 megohms. Also excluded wells where the holes were probably blocked. This was in where wells having greater than 500 megohm resistance. And we also tried to refine it even further by looking at the quality of the seal, actually looking at the trace of the seal here, and actually taking the capacitance transient peak and looking at a ratio in terms of the peak to the current plateau from the voltage step during the seal test. And we set an arbitrary threshold of 0.5, but this helped us exclude some wells, further wells that probably where there wasn't a quality seal, and helped increase our Z prime and our quality of the data that we were getting from these assays. So again, this was just an arbitrary threshold that we set, but the 0.5 seemed to work well for us initially. Also, again, once we exported the seal test and the compound trigger, the actual traces, and pulled out the quality traces, we did some manipulation of these traces in which we would remove the tip touch artifact, which is shown here on the upper right-hand side where the cursor is. We would also take the no compound control traces. That was our from column 1, in which no compound was added. And we would take those control traces and subtract that from our current traces, and normalize the baseline, and then export this file as a cleaned up text file as an Axon text file that we can open it with the pclamp software for any type of further analysis. once we had the cleaned up traces, the quality traces were poor seals were excluded, and the no compound control was subtracted. We then matched this up with a Barracuda plate map, plate load map that was designed for each experiment that we ran, and this was just a template form in Excel, and this highlights what compound and what concentration are in each well. This Excel plate map was referenced with the compound traces so that it would give us this particular output right here. This algorithm took anywhere from 3 to 5 minutes to run, and once we exported the raw trace and seal test data and had the plate map, it would export all of the dose response curves for each compound and for each metric again that we found was valuable. So primarily I'll be showing amplitude data. In some coming slides I'll show you the decay constant dose response curve as well, and tell you why we think that might be important in some of the compounds that we were investigating. So jumping into the validation data from this study, again we were looking at comparing across 3 different platforms responses from the alpha-4 beta-2 and the alpha-3 beta-4 nicotinic receptor. And just to orient you here, on the left-hand side, the Dynaflow system is our manual patch clamp system. The Patch Express is, again, that medium throughput automated system which can achieve the giga-ohm seals. And the Barracuda is, of course, again, the high throughput automated system with the perforated loose patch system. So checking out across the different platforms here, we'll start with the alpha-4 beta-2 receptor. And again, this receptor has been shown to exist in 2 different stoichiometries, having both a high sensitive and a low sensitive component. So we can do a biphasic fit to the dose response curve, and as you can see, example traces here from our manual patch clamp experiments, along with the EC50 for both HS, the high sensitive, and the LS component. And as we track that across to the Patch Express with both the HS and the LS, look at the example traces, and then across to the Barracuda, we see a nice consistency with the biphasic fit and with the 2 high sensitive and low sensitive EC50 values as shown here. Similar data shows for the alpha-3 beta-4 receptors. Again, this is just a single exponential or Hill equation fit for the dose response. Again, this is all amplitude data, and we can track it along here with the example traces in the EC50 from the manual Dynaflow system to the medium throughput automated system to the higher throughput Barracuda system, and we see a nice comparison across all 3 platforms, a nice consistency for the EC50 data. So we feel that this was a nice validation of the Barracuda system for looking at agonists for the alpha-4 beta-2 and the alpha-3 beta-4 nicotinic receptors. We also were interested in looking at antagonism at these receptors as well, and we looked at both. We wanted to investigate both a competitive antagonist and an uncompetitive antagonist, and just to sort of walk through the differences there. So we're all familiar with the competitive antagonist. The compound will bind at the orthosteric site and inhibit the ligand from activating the channel. There's also the non-competitive antagonist, which binds at a separate site, an allosteric site, and inhibit the channel from being opened or activated. And we were also interested in uncompetitive antagonism, and this is the type of antagonism where it's contingent upon prior activation of the channel, so that the channel needs to be activated and opened for an uncompetitive antagonist to inhibit the current. And this is typically seen as a use-dependent or open channel blocker. And that's, Dr. Roth had a nice explanation of the state-dependent versus state-independent inhibition of the sodium channel in the first presentation. And I think the competitive or non-competitive antagonism versus the uncompetitive antagonism, it's a nice is analogous to what he was talking about there, where the inhibition is dependent upon whether or not the channel has to be activated or not. So we first looked at competitive antagonism in the ionworks-Barracuda system. And we were looking at alpha-4 beta-2 nicotinic receptors and investigating the prototypical competitive antagonist here, DH-beta-E. And as you can see in our Dynaflow system, looking at DH-beta, the dose effects of DH-beta-E to inhibiting current produced by 10 micromolar of ACH. You can see the nice curves here on the upper right-hand portion of this plot, and the dose response for the DH-beta-E inhibition of acetylcholine. When we looked at this in the Barracuda, again, we saw a nice dose response for inhibition of the response to 30 micromolar of acetylcholine with an IC50 value here, so around 600 nanomolar for the Amplitude. And again, this is here we looked at a different metric. So we not only looked at the amplitude, which is shown here where my cursor is, but also the decay constant. And so again, we just fit an exponential function to this portion of the aggregated current response right here, and did a dose response. And again, showed a nice dose response of inhibition with DH-beta-E. And these two curves from the peak amplitude and the decay constant look fairly similar. There's not much of a difference between the IC50, maybe a fourfold difference between these two. And so it appears that this competitive antagonist would have a similar effect not on the amplitude, the peak response, but also on the decay constant as well. We also were interested in, as I mentioned, the uncompetitive antagonism. And here, this has been shown with manual patch clamp recordings, how you actually tease out an uncompetitive antagonist. This is done, and actually it's also been done with sodium channels as well, sort of that really elegant slide that Dr. Rock had on the pulse train, looking at the first versus the 25th pulse. We do something similar in manual patch clamp, where we'll have multiple applications of the endogenous ligand of acetylcholine, 10 applications showing this use-dependent inhibition of mecamylamine, which is an uncompetitive antagonist to alpha-4 beta-2. So in the presence of mecamylamine with repeat applications, you can see this nice use-dependent inhibition of the current response over time with the different concentrations of mecamylamine used. When we plot the dose response for the first pulse, which is shown here, versus the 10th pulse, which is shown here, you again see that there's a much more potent inhibition at that 10th pulse, shown where the 787 nanomolar IC50 on the 10th pulse as compared to the first pulse, which is a less potent 15 micromolar IC50. Now, when we did this in the Barracuda using the uncompetitive antagonist mecamylamine on alpha-4 beta-2 receptors in the ionworks Barracuda, we saw something very similar, which was interesting to us. So when we looked at the peak amplitude, the effect that mecamylamine had on the peak amplitude of the response, the aggregated current response in the Barracuda system, the traces are shown down here, the effect on this peak amplitude. It wasn't that great. It was a very potent, not very potent inhibition with an IC50 of around 30 micromolar on the peak amplitude. But when we looked at the decay constant, and it's pretty evident by the traces here, you can see that it increases the rate of decay, or it quickens the decay of the current response much more potently. So when we do a dose response curve with mecamylamine to the normalized response of the decay constant, we can see that it's a much more potent IC50. And this degree of separation between these two is fairly similar to what's seen on the first pulse versus the 10th pulse over here on our manual patch clamp experiments. And so this led us to conclude that what we're co-applying both acetylcholine and the uncompetitive antagonist mecamylamine, and it seems to have a much greater effect on the tail part of the aggregated current response. So we think that there are multiple activations of the channel over that 8-second co-application of the agonist and the antagonist, and it has a much greater blocking effect on, it has a much more potent effect of blocking repeat openings or multiple openings over that time course. And so we feel that this is analogous here, looking at the peak amplitude and decay constant, analogous to looking at the first pulse versus the second pulse in our manual patch clamp experiments. We saw similar effects on the alpha-3 beta-4 receptor as well, looking at the mecamylamine, the competitive antagonist, again showing the manual patch clamp experiments on the left-hand side. Again, a nice use-dependent inhibition as the concentration of the agonist increases. Again, so we're looking at number of ACH applications across the bottom here in the presence of mecamylamine, the uncompetitive antagonist, and as you increase the number of applications, you get this nice use-dependent inhibition. And we also, again, plotted the first pulse versus the 10th pulse on a dose-response curve, and again saw a much more potent inhibition at this 10th pulse versus the first pulse. And that also, again, bore out with the experiments done on the Barracuda, where in the presence, the co-application of both acetylcholine and mecamylamine on the response, the alpha-3 beta-4 response, we see that it's not as potent on the peak amplitude as measured here, but is much more potent on the decay constant, as you can see on these example traces here. And so again, this degree of separation between the amplitude and decay constant IC50s, very similarities to the IC50 discrepancy shown between the first pulse and the 10th pulse in our manual patch clamp recordings. And so, yeah, with that, I'm going to conclude and wrap up the slide presentation. But just to kind of summarize what we showed is, and despite of the differences in methodological approaches between manual patch clamp recording on the Dynaflow system and automated electrophysiology on the Patch Express and ionworks systems, which have differences again in seal quality, giga-ohm seal versus the loose perforated patch seal. The EC50 values at both alpha-3 beta-4 and alpha-4 beta-2 nicotinic receptors were relatively well preserved and showed nice consistencies across all three platforms. In addition, we validated the ionworks Barracuda for both competitive and uncompetitive antagonist assays at the alpha-3 beta-4 and alpha-4 beta-2 receptor. So we feel that looking at both the dose response curves for amplitude and for the current decay, or the decay kinetics of the aggregated response, could be a good indicator of whether or not we're seeing a competitive or an uncompetitive antagonist in our assay. And that being able to do that, to compare peak amplitude versus decay kinetics of the aggregated response, we're able to do that by using our offline analysis of the raw exported data with our custom written MATLAB function that can rapidly generate these concentration response curves and the desired metrics that we weren't able to perform using the built-in Barracuda analysis software. So we've really optimized this assay and analysis paradigm for both agonist and competitive and uncompetitive antagonist assays. And with that, I just want to finish by making a few acknowledgements to the Target Steps former Electrophysiology group. Nikolai Fedorov, Sergey Sabach, Hai Young-Wei, and Lisa Benson were all former members of our electrophysiology group here at Target Steps. So with that, I'll conclude my presentation.
Thank you, John. Very nice looking data. So we'll have a short question and answer period here. Please submit your questions using the Q&A tab or the chat tab. And John, since we have you live, why don't we go ahead with a couple questions for you first?
Okay. you want me to pass the ball back to you, James?
Yeah, I just
okay. Okay.
The first question is, hold on, let me just share my application. First question is, can you comment on the validity of using kinetic data from the aggregated current response in light of the loose fields achieved with the perforated patch on the ionworks-videos?
Yes, I think this is an important point. So we're not really saying much. We can't really say anything definitively about the effects on individual single-channel kinetics. Obviously, with the perforated patch and the loose field quality, we don't have the types of fields that we can really get a good idea of the kinetics of channel responses. I think using the aggregated response kinetics, the decay constant is a good primary indicator of competitive versus uncompetitive. You can't get any definitive numbers on kinetics, but I think it's a good would be a good comparison to help categorize potential antagonists as either competitive or uncompetitive, and offers that type of primary screening distinction rather than any sort of comment on the effects on channel kinetics.
Okay, thank you for that. And also, have you ever tried alpha-7 nicotinic receptors?
Yeah, so we have, and alpha-7s are a little trickier. They are a very fast desensitizing receptor. The kinetics of those receptors are much faster than the alpha-4 beta-2 and alpha-3 beta-4, and so we have looked at those receptors in our assays. We've had to play around with the height of compound in terms of which we add the compound, and also the speed at which we add. So I think lower and slower seems to work best for those receptors. And if I'm not mistaken, Jim, you can probably correct me if I'm wrong. I think Jim has does he have a webinar on some alpha-7 data that he collected?
Yeah, he has not presented that. We do have some data, but we haven't presented that yet.
Okay, so I know we haven't presented it either. We've been trying to optimize it, but we have tried it. We have had some successful recordings, but we haven't presented that data.
Okay, thank you. So I think we'll take a few questions for Dave now. So Dave, I'll unmute you here. Are you there, Dave?
Yep, I'm here.
Okay. So, question for you. The IV curves and raw data traces for activation of sodium 1.8 look well behaved in terms of voltage clamp control. Is this the case for sodium 1.5 and 1.7 that normally have higher expression levels?
Well, yeah, it's a good point. I was a bit surprised to see how well behaved the IV curves look like for NAV 1.8. And I really think that does have something to do with the smaller amplitudes of current. Because as everyone knows, the ionworks platforms, both Quattro as well as Barracuda, there's no series resistance compensation. So when we get extremely large currents, we start to see a much steeper IV curve. And I presented back in 2009 at one of the automated users meetings around NAV 1.5 on Quattro, and when we did manual recordings and turned off series resistance compensation, our recordings in the manual setup for NAV 1.5, which the expression level is usually much higher, 4 or 5 times higher than what we have with 1.8. The manual recordings without series resistance compensation looked exactly like the Quattro recordings, and that's what our 1.5 and 1.7 currents look like.
Okay, thank you. Another question for you is, you indicated that obtaining high expression of sodium 1.8 is a challenge. How many patches can a cell line tolerate before there are issues with the expression?
Well, one of the things that we haven't systematically studied that, but with our NAV 1.8 line, we've kept it in continuous culture for over 2 months, which are probably 15 to 20 passages, without seeing significant loss of expression or any issues with fields in the Barracuda. We haven't tested anything really beyond that, because our standard practice is not to keep cells in continuous culture for more than 2 months. And in fact, with our other sodium assays, as well as 1.8, we have high density frozen plugs that are in the range of 10 million cells per plug. And we bring those out and they can be ready to test in the ionworks platforms in a day or within a few days of when we thaw them. And we usually get good expression with them right off the bat. There's no kind of ramp-up period with lines that we've currently been working with. So it is a challenge, and I would suggest that most people that use the ionworks platforms try to keep down to low passage numbers, and I think this high-density frozen cell plug is a great way to go in terms of really being able to run assays with good reproducibility. We do the same thing with our herd cells, and it's made a tremendous difference in terms of lowering our run-to-run variability.
Okay, thank you. Certainly, frozen cells have come a long way in the last 5 years. One more question for you, Dave. The two-side fit of the Tetracaine data in the twin pulse inactivation protocol is interesting. Do you see a similar profile with shorter recovery delays?
We do, and we probably looked at it more in the 200 millisecond than some of the other short delays, because the current amplitudes get down fairly low with the very short recovery delays. We do see a component that's that low affinity component that was 33%, I think, if I remember correctly now, of the block. That is smaller with the shorter delays. So it drops down to about 15 to 16% and gets hard to fit. You can see it in the data. It gets harder to fit with the shorter delay. Very interestingly, we have done the same kind of protocols with NAV 1.7, and I looked into the data specifically at the 100 and 200 millisecond delays, and we don't see that second site. It's a single site fit in the NAV 1.7 data, suggesting that there's something about the biophysics of NAV 1.8 that makes it different from 1.7 with those compounds. That's something we're going to look into more, but it's kind of interesting.
Yep. Thank you, Dave. So we are still getting a lot of questions through the chat and the Q&A tab. So what we'd like to do is we'll answer the remainder of the questions by email, to your personal email. We'll leave the line open for 5 minutes here so you can continue to send in questions. But I think what I'd like to conclude the webinar here, or the speaking portion of the webinar, and thank presentations. And with that, I think we're going to go ahead and go silent, and we'll leave the line open for 5 minutes to collect more questions. Thank you, everybody, for attending.